The key to life is nucleotide formation. Nucleotides are the building blocks of DNA and RNA, the information storage molecules of our genes.
RNA is ribonucleic acid. DNA is deoxyribonucleic acid. Both molecules are long strings of nucleotides. Nucleotides are the building blocks of DNA and RNA. Nucleotides are composed of three smaller parts: a phosphate, a ribose sugar, and a nucleobase. A phosphate is just a phosphorus atom surrounded by 4 oxygen atoms, and so has the chemical formula PO4. Phosphate is a naturally occurring inorganic molecule, pretty common on Earth now, and likely common on a prebiotic Earth. There is little mystery on how nucleotides obtained phosphate naturally. There is much more mystery on how the ribose sugar and nucleobases originally formed in the prebiotic oceans, or in an isolated prebiotic pond on an ancient, lifeless Earth. Since the discovery of the DNA double helix structure by Watson and Crick in 1953, Origin of Life researchers have been trying to solve the mystery of how the deoxyribonucleic acid nucleotides were formed in the prebiotic ocean or prebiotic pond where life might have originated. In modern cells, protein enzymes assemble phosphates, ribose sugar, and nucleobases into nucleotides. These building blocks themselves are synthesized by other protein enzymes. In a prebiotic world, none of these protein enzymes would exist. So, how did nucleotides and their building blocks come to be synthesized in a world before proteins? In 2009, a team of chemists led by University of Manchester, England, Professor John Sutherland, published a paper that gave a plausible chemical pathway of how nucleotides might have been synthesized before proteins. In 2009, Matthew Pauner, Beatrice Gerland, and John Sutherland wrote a paper titled Synthesis of Activated Pyrimidine Ribonucleotides in Prebiotically Plausible Condition. The paper was published in May 2009 in the British science journal Nature. That 2009 paper was accepted and celebrated by the Origin of Life community as the first plausible process by which nucleotides may come about naturally on a prebiotic Earth. For this video, I will analyze the results of the 2009 Pauner, Gerland, Sutherland paper. In 2009, the Pauner and Sutherland team described their initial study of nucleotide synthesis in a scientific paper published in the journal Nature. In 2015, Sutherland did an expanded study with a different team. The 2015 Patel-Sutherland paper from the 2015 study focused more on amino acid synthesis. Figure 1 of the 2009 paper gives just the nucleotide synthesis pathway. In Figure 1 of the 2015 paper, the chemical system is expanded to include the amino acid pathways. Figure 1 of the 2009 paper is embedded in the section A, or the light blue areas of Figure 1 of the 2015 paper. The chemical species are renumbered between the 2009 and 2015 papers. But the chemical species in blue areas of Figure 1 the 2015 paper are the same as the chemical species of Figure 1 of the 2009 paper. On the direct pathway to Sutherland's prebiotic nucleotide synthesis process are 2 other prebiotically derived chemicals, namely glycolaldehyde and cyanamide. In Figure 1 of the 2009 paper, glycolaldehyde is species 10, and cyanamide is species 8. Glycolaldehyde, C2H4O2, is a precursor for sugar. Glycolaldehyde can be obtained naturally through the Formose reaction acting on naturally occurring formaldehyde. Cyanamide, CnNH2, can be obtained through a multi-step process starting from naturally occurring ammonia, NH3, and naturally occurring common lime calcium oxide, CaO. Glycolaldehyde is the first of the starter molecules for nucleotide synthesis. Cyanamide is really a man-made substance. The natural formation of cyanamide could be possible only if a series of steps are followed at high temperatures. Cyanamide NH2CN is the second molecule that Sutherland needs to start his nucleotide synthesis process. I can't tell you what the concentrations of cyanamide or glycolaldehyde would be in the prebiotic ocean or a small prebiotic pond. These chemicals don't exist in more than trace concentrations in small ponds today. I don't know about the prebiotic concentration of glycolaldehyde. Call me skeptical about the presence of cyanamide. Let's just assume, along with Sutherland, that these chemicals did exist in sufficient concentrations in a small pond to start Sutherland's nucleotide synthesis process. In the 2009 paper, Sutherland shows a plausible pathway from which nucleotides can be generated from cyanamide and glycolaldehyde. According to Sutherland, both of these starting chemicals would likely be available on a lifeless Earth in prebiotic times. Earlier attempts by researchers to naturally synthesize ribose sugar and nucleobases separately have failed, especially for the ribose synthesis. Ribose sugar is terribly difficult to synthesize without modern lab equipment and modern reagents that would not be available on a prebiotic Earth. Sutherland's process bypasses the separate synthesis by forming ribose and nucleobases together on the same intermediate product. Sutherland claims good results: a high yield of cytosine and uridine nucleotides, over 40%. The 2009 Sutherland paper outlines the pathway to nucleotides in Figure 1 of that paper. In the figure, glycolaldehyde is labeled as species 10, and cyanamide is listed as species 8. The end goal, a cytosine analog nucleotide, is labeled as species 1. Figure 1 outlines two pathways, a blue path and a green path, to get from starter species 8 and 10 to end goal species 1. The blue pathway was found to fail when species 3 and 4 could not produce species 2. The blue pathway is marked with a red X at the point of failure. Along the blue path, the glycolaldehyde species 10 rearranges into intermediate species 9, glyceraldehyde. The triple carbon C3HN or HCCCN. Species 7 could come from another process. On the successful green pathway, the glycolaldehyde and cyanamide react to form intermediate species 11, a precursor nitrogenous base. Along the green path, intermediate species 9 and 11 react to form intermediate species 12. Species 12 is called arabinose amino-oxazoline. The arabinose part of species 12 is a pentose sugar in the same pentose family as the ribose sugar needed for an RNA nucleotide. Species 12 reacts with the waiting species 7 to become species 13. Species 13 is called anhydroarabinonucleoside. Sutherland assumes the green pathway occurs in an aqueous water solution buffered by phosphate PO4. So, once species 13 forms, it reacts with the phosphate buffer to become species 1. Beta-ribocytidine 2'3' cyclic phosphate, an analog of the RNA nucleotide cytosine. When we compare species 1 to the regular structure on the cytosine molecule, we see that the phosphate group is attached at the wrong carbon on the ribose sugar. We can give it to Sutherland that at least some of species 1 could become rearranged so that the phosphate groups will be in the proper position to become proper cytosine nucleotide molecules. Success. at least on paper. As we read the paper more closely, there must be a drying step between species 13 and species 1. From what I understand, each of the steps in Figure 1 were done separately using purified chemicals to start. The first intermediate yield is recorded. For the next step, brand new purified first intermediate is used in fresh, clean experiment, and the yield of the second intermediate is recorded. So we never get to see the yield of nucleotide species 1 from reactions of the starter species, 10-glycolaldehyde, and species 8, cyanamide. They start with purified chemicals for every step. This purified step process avoids the buildup of all the waste intermediates and waste side reaction products that would clog up the process pathway from starter species to nucleotide. The purified lab step is not what would occur in nature, where all the side reaction waste might hinder a good yield. In Figure 2 of the 2009 Sutherland paper, the research team is just looking at the first step. What is the yield of intermediate species 11 when species 8 and 10 are allowed to react to each other? Species 8 is cyanamide. Species 10 is glycolaldehyde. Sutherland reports this result as a greater than 75% yield of species 11 when species 8 and species 10 are mixed in a 1-to-1 ratio. That result is pretty good yield for the first step in the process. In Figure 2b, we see all the intermediate products and side products that lie on the pathway between the mixing of species 8 and 10 and the goal intermediate species 11. The reaction occurs in the absence of the phosphate buffer. We see that there are 6 other intermediate products between species 8 and 10 and the goal intermediate species 11. There are probably other products that do not appear in 2b. I think a major takeaway from Figure 2b is that the process shown in Figure 1 is greatly simplified. In Figure 2d, we see the influence of the phosphate buffer on the cyanamide species 8 on its path to form intermediate species 6 urea. Figure 2d shows that the cyanamide species 8 form intermediate species 19, and species 19 has a divergent path into both urea species 6 and cyanoguanidine species 20. In 2009, Sutherland Figure 3, we see the side products and yields of 3 reactions. Figure 3a shows chemical structures other intermediates and side products that might form alongside Figure 1 species 12. Sutherland needs intermediate species 12 for the Figure 1 pathway between the starter cyanamide species 8 and starter glycolaldehyde species 10 and the end goal nucleotide species 1. There are 6 other species formed alongside species 12. Sutherland calls species 12 and species 21 the major products of mixing pure species 9 into pure species 11. The results of this reaction were allowed to rest for a full day to see how the side products stabilize in the phosphate buffer solution. Figure 3b shows that, in isolation, product 12 will separate into product species 12 and species 24. After a full day, the stabilization test results in 56% species 12 and 16% species 24. Percent yields of other stabilization products are not listed. I tabulate the results of the Figure 3b stabilization tests here. Finally, Figure 3c shows the result of a 2-step process starting with cyanamide species 8 and glycolaldehyde species 10 to produce species 11 in the first step, and then in a second step. A sample of species 11 is removed, placed in a different beaker, to which species 9 is added until species 12 is produced along with the other side products. I quote from the original 2009 paper. Quote, "We then took a crude sample of 11 that had just been prepared from cyanamide 8 and glycolaldehyde 10 in the presence of phosphate, and added glyceraldehyde 9 to it. After overnight incubation, HNMR analysis revealed that although all 4 amino-oxazolines were still formed, the lixose derivative 23 was selectively depleted and was now a minor product along with the xylose derivative 22, figure 3c." Unquote. So, by this quoted procedure, after letting the mixture react and stabilize overnight, the mixture is analyzed to get the percent of wanted species 12 produced, along with the unwanted co-products. According to figure 3c, the reaction yield of species 12 is 15%. The yield of species 21, 22, 23, 24, and 25 are 25%, 6%, 4%, 11%, and 9% respectively. These percent yields are listed in figure 3c. So, if we go back to figure 1, we might get 15% of 80% of the original glycolaldehyde species 10. Or, if we disregard that species 9 was added later, we get 12% yield for species 12. In a natural prebiotic reaction, species 9 could not be added later, and all the waste products of the species 11 production would still be present to limit the yield of species 12. In the 2015 Patel, Sutherland et al. Follow-up paper, Sutherland proposes his separate beakers could represent a network of pools along a stream that would allow separate reactions to occur at different locations and times. Moving on to figure 4. In figure 4 of the 2009 Sutherland paper, we see the result of isolating species 12 from the other products. For figure 4a, Sutherland isolated species 12 and placed it in an aqueous solution with cyanoacetylene species 7. The unbuffered result yielded 56% beta-arabinocytidine species 26. Beta-arabinocytidine is not a nucleoside because arabinose is not ribose, despite the fact that both arabinose and ribose are pentose sugars. Arabinose does not pucker the way ribose does, so an arabinose-based nucleotide would not form the stable spiral of RNA or the double helix of DNA. I covered the importance of ribose puckers in my video on ribose sugar. Figure 4a also shows the result when isolated species 12 was placed in solution with cyanoacetylene species 7 in a phosphate buffered solution. The buffered result is a 92% yield of the desired arabinose anhydronucleoside species 13, along with co-products of 16% cyanovinyl phosphate species 27 and 1% beta-arabinocytidine species 26. Pay attention to species 27 cyanovinyl phosphate, as it will play a special role in procedure A of figure 4b. As species 13 is on the pathway shown in figure 1, the high 92% yield of species 13 is very promising for production of nucleotide species 1. The downside to the experiment is that Sutherland had to isolate species 12. It is unlikely that species 12 would be isolated in nature. Sutherland's solution to this isolation problem is that products were produced in isolated pools along a stream, and only the needed species for the next step was carried by the stream of water into the next isolated pool. Up to and including figure 4a, all the reactions happen in water. In an aqueous solution, water is the solvent. Figure 4b is where the pathway goes off the rails into the realm of faith and chemistry. In figure 4b, there are two procedures, procedure A and procedure B. Neither of these procedures take place in water. Procedure A is dry. No solvent. According to Sutherland, while it is still in the water, aqueous solution, the species 27 cyanovinyl phosphate formed in Figure 4a reacts with the inorganic phosphate buffer to form pyrophosphate. So, we are expected to have faith and believe that for Procedure A to come about in nature, that the cyanovinyl phosphate species 27 of Figure 4a is converted into pyrophosphate, and then pyrophosphate and the arabinose anhydronucleoside species 13 leave the other coproducts behind and are transported in isolation downstream into a separate pool. That separate pool must then dehydrate and dry up so that the procedure A dry reaction can happen. In Figure 4b Procedure B, the solvent is formamide, not water. So, again, we are expected to have faith and believe that the arabinose anhydronucleoside species 13 of the 4a reaction leaves the other coproducts behind and is transported in isolation downstream into a separate pool. That separate pool must then dehydrate and dry up. That separate dry pool must then be filled with the solvent formamide, so that the Procedure B reaction can happen. I understand that natural pools and streams may dry up every year during the dry season. That such a dry pool would then fill with formamide before the next rain is a stretch of my imagination. In Figure 4b, we see the result when the two procedures are applied to isolated arabinose anhydronucleoside species 13. In Procedure A, dried species 13 is added to dry urea species 6 in dry pyrophosphate. The result of Procedure A is a yield of 32% of the cytosine nucleotide species 1, along with a trace of species 3, 17% species 13, 2% species 26, 7% species 28, 14% species 29, 2% species 30, and a trace of species 31. These percent products are shown for Procedure A in Figure 4b. Urea species 6 is formed from cyanamide species 8 in Figure 1. Urea species 6 is reasonably available in nature. For Procedure A to occur in nature would assume that isolated arabinose anhydronucleoside species 13 dried out in an isolated pool along with urea and pyrophosphate. That pool's neither hydrogen cyanide nor formamide solutions are very common on modern Earth. Your guess is as good as mine as to whether Sutherland's theory of nucleotides being formed in a formamide solution is a good theory. Maybe nucleotides forming in a formamide solution is plausible, as Sutherland suggests. Maybe not. It depends on your faith that formamide could condense into a dry pool containing arabinose anhydronucleoside urea and inorganic phosphate. It's easy for a chemist to mix these ingredients in a lab. It's far less likely to happen in nature. This improbability makes the formation of nucleotides by this process a secular miracle, if not a supernatural miracle. 18% yield of species 31. These percent products are shown for Procedure B in Figure 4b. As stated, urea species 6 is reasonably available in nature. Formamide can be the natural result of a couple of natural processes. What is not natural is that arabinose anhydronucleoside, urea, and inorganic phosphate would end up in a waterless pool filled with formamide solvent. It's easy for chemists to mix these chemicals together in a laboratory. It's an act of faith for these chemicals to end up together in nature on a prebiotic Earth. In Figure 5 of the 2009 Sutherland paper, Sutherland shows how the analog cytosine nucleotide species 1 partially converts into an analog uridine nucleotide species 33 after 3 days of exposure to ultraviolet radiation UV. The exposure of species 1 to 3 days of UV light yields 43% cytosine nucleotide species 1, 42% analog uridine nucleotide species 33, and 5% cytosine nucleobase species 3, and 4% uracil nucleobase species 34. Sutherland says this 3 days of exposure to UV light destroyed many of the co-products that might interfere with the incorporation of the nucleotide products into RNA. If possible or potential is what they mean by plausible, I agree that Sutherland and company have discovered a possible pathway or a potential pathway for natural nucleotide synthesis. I know the 2009 paper and follow-up Sutherland papers have been celebrated in the Origin of Life research community. I can't argue that the isolated existence of arabinose anhydronucleoside species 13 and urea species 6 in formamide solvent buffered by inorganic phosphate, then irradiated by UV light, is not predisposed to react and form nucleotides. It's not just predisposed, it's definite. As Sutherland and company demonstrated in Procedure B, both cytosine and uridine nucleotides will definitely form with greater than 40% yield under the conditions of Procedure B plus UV irradiation. What is not predisposed is that arabinose anhydronucleoside species 13 and urea species 6 will ever come to exist in isolation in a natural prebiotic setting. Further, is it even likely that arabinose anhydronucleoside species 13 and urea species 6 will ever come to exist together naturally? Sutherland and other Origin of Life researchers think it's plausible. Skeptics like me think that is unlikely that arabinose anhydronucleoside species 13 and urea species 6 would ever come to exist together naturally. I would go further and say that the only likely way that arabinose anhydronucleoside species 13 and urea species 6 ever came together in formamide solvent with inorganic phosphate would be because an intelligent chemist like Sutherland or some supernatural intelligent designer put those 4 chemicals together. It would be impossible to calculate the Las Vegas odds that 4 obscure chemicals could come together in some isolated pond on prebiotic Earth. We don't have enough information. We don't know the abundance of any of these 4 chemicals on a prebiotic Earth. Of the 4 chemicals needed, only the inorganic phosphate is a naturally occurring substance. Inorganic phosphate occurs naturally around only a few phosphate mines around the world. Most phosphates found in nature are the result of biological processes. Urea species 6 is a simple enough chemical to make naturally. Figure 1 of the 2009 Sutherland paper shows urea species 6 as a product of cyanamide species 8. We don't know the abundance of cyanamide on prebiotic Earth. Urea, CONH2 could easily be formed by exposing a mixture of carbon dioxide CO2 and ammonia NH3 gas to lightning. The third substance, formamide HCONH2, was less likely to be present on a prebiotic Earth. Or, formamide could come to be from a reverse reaction of hydrogen cyanide HCN with water H2O. HCONH2 becomes HCN plus H2O. So, maybe formamide was present on a prebiotic Earth. I can't find a single pool of formamide on the modern Earth. Of course, there may be a bias in my modern thinking. So, phosphate is naturally occurring in certain small locations. Urea and formamide are relatively easy to form in a carbon dioxide and ammonia atmosphere with lots of lightning. I don't know what the Las Vegas odds of having 3 out of 4 ingredients exist in an isolated pool. Maybe it was possible. Maybe plausible. The arabinose anhydronucleoside species 13 is a bigger problem. We need that fourth ingredient to get a legitimate nucleotide. This fourth ingredient is the most important, because without arabinose anhydronucleoside species 13, the other three more natural ingredients are not going to matter. So, what are the Las Vegas odds of getting that fourth all-important ingredient, species 13, isolated in the soup with the phosphate, urea, and formamide? I don't know how to figure the odds either. There is not enough information. I feel the odds must be high. Possible, but not likely. Let's remember that arabinose anhydronucleoside species 13 is the end product of a multi-step process that start with cyanamide species 8 and glycolaldehyde species 10. And the starter species must exist in a totally separate pool. Upstream of the pool where the arabinose anhydronucleoside species 13 must end up in an isolated pool with the phosphate, urea, formamide ingredients. To make this all work naturally on prebiotic Earth, we must have a system of pools interconnected by streams that flow downhill into the next pool below. We must redraw Figure 1 with the pools named for the ingredients that must form in each pool. This system of pools is how Professor Sutherland explains it in his follow-up papers. Sutherland's 2015 paper, with collaborators Patel, Percivalle, Ritson, and Duffy, is titled Common Origins of RNA, Protein and Lipid Precursors in a Cyanosulfidic Protometabolism. The 2015 paper specifically mentions a network of pools connected by streams. Pool 1 would be the lowest pool where the reaction shown in Figure 5 occurs. The cytosine nucleotide species 1 is irradiated by UV for 3 days to stabilize into cytosine nucleotide species 1 and uridine nucleotide species 33. Pool 2 would be higher and upstream of Pool 1. This Pool 2 has the ingredients arabinose anhydronucleoside species 13, urea species 6, and pyrophosphate buffer. All the ingredients of procedure A of Figure 4b. Alternatively, this Pool 2 has the ingredients arabinose anhydronucleoside species 13, urea species 6, formamide solution with an inorganic phosphate buffer. All the ingredients of procedure B of Figure 4b. In Pool 2, the cytosine nucleotide species 1 is produced. If formamide is to be the solvent in Pool 2, there must be a Pool 3 in which the formamide condense upstream of Pool 2. The formamide drains into Pool 2 once it dries out. Upstream of Pool 2, there must be a separate Pool 4 that is the source of urea species 6. Urea is formed from carbon dioxide CO2 and ammonia NH3. Also, upstream of Pool 2 is Pool 5. In Pool 5, there is the Figure 4a reaction, in which species 12 and species 7 react into the arabinose anhydronucleoside species 13. Pool 5 is a water aqueous solution containing an inorganic phosphate buffer. The network of pools leading into Pool 2 is shown. The species 12 and species 7 for Pool 5 must have formed in their own separate pools upstream of Pool 5. Species 7 is cyanoacetylene, C3HN or HCCCN. Cyanoacetylene species 7 is a 3-carbon molecule. Cyanoacetylene species 7 was formed in the famous Miller-Urey experiment, where methane CH4 and ammonia NH3 were energized by a spark to represent lightning passing through the prebiotic atmosphere. So, cyanoacetylene species 7 could have formed naturally in its own separate pool. Pool 7, from a reaction of carbon dioxide CO2 and ammonia NH3. Species 12 is formed in Figure 1 from a reaction of species 9 with species 11. Let's form species 12 in pool 6. Figure 3c shows that species 12 could be formed in the same aqueous solution as species 11. That was formed from cyanamide species 8 and glycolaldehyde species 10. For consistency, let's put species 11 in its own pool, pool 8. Glyceraldehyde species 9 would have to form from glycolaldehyde species 10 in its own separate pool, pool 9. The primary sources species are in their own separate pools that feed the network intermediate pools as necessary. Pool 10 contains the glycolaldehyde species 10. Pool 11 contains the cyanamide species 8. Pool 7 contains cyanoacetylene species 7. As we can see from the network, Pool 7 and Pool 6 feed Pool 5, where the arabinose and hydronucleoside species 13 is formed. Pool 8 and Pool 6 could be the same pool, but we keep them separate for simplicity, and because the species 11 must form before Pool 9, species 9 is needed to form species 12. Pool 10 contains the glycolaldehyde species 10. Pool 10 feeds glycolaldehyde species 10 to both Pool 9 and Pool 8. In Pool 9, glycolaldehyde species 10 is converted into glyceraldehyde species 9. In Pool 8, the flow of the Pool 10, glycolaldehyde species 10, mixes with the flow of cyanamide species 8 to form the species 11 intermediate formed in Figure 2b. I want to question the availability of the primary chemicals, glycolaldehyde species 10, cyanamide species 8, and cyanoacetylene species 7 on a prebiotic Earth. The Pool 10, glycolaldehyde species 10, is the result of the Formose reaction that converts formaldehyde to glycolaldehyde under UV light exposure. Yes, formaldehyde is a naturally occurring chemical species, but the Formose reaction to create glycolaldehyde species 10 is slow. I question whether the natural supply of glycolaldehyde species 10 is enough to supply both Pool 9 for the formation of glyceraldehyde species 9 and Pool 8 for the formation of intermediate species 11. The Pool 11, cyanamide species 8, is also hard for nature to supply. Cyanamide CnNH2 can be obtained through a multi-step process starting from naturally occurring ammonia, NH3, and naturally occurring common lime calcium oxide, CaO. These multi-step processes are natural, but they require high energy. I agree the heat could be supplied by lightning or volcanic heat. We are asking a lot from nature to supply us with difficult-to-obtain chemicals, natural or not. For Pool 7, cyanoacetylene species 7. I already said that cyanoacetylene could form naturally from lightning, passing through the prebiotic atmosphere of methane CH4 and ammonia NH3 and CO2. So, I have no doubt that cyanoacetylene species 7 could have formed naturally on its own. I just question the quantity of cyanoacetylene species 7 needed to fill Pool 7, so that it in turn can feed Pool 5. So that intermediate species 12 and cyanoacetylene species 7 can react to make arabinose and hydronucleoside species 13. It is pretty easy for Sutherland and his colleagues to get the right quantities of all these source chemicals in their well-stocked laboratory and fill their cleaned experiment beakers for the next reaction. It is a lot harder to expect nature to fill the source pools with the right chemicals at the right time, and then transfer the right intermediate in the next pool in the network so that the next intermediate species is formed. Then we get to Pool 5, where the arabinose and hydronucleoside species 13 is formed. Nature has to wait for the pool to dry up, or for the water solvent to be replaced by formamide solvent. So the reaction from arabinose anhydronucleoside species 13 into cytosine nucleotide can take place. All these steps, and all these separate pools where separate reactions are taking place, is a lot to ask from nature. It's a long shot in gambling terms for all these steps to happen naturally and in the correct order. Sometimes the long shot horse does win. I can't deny that. It is certainly not predisposed for nucleotides to form. It's like getting snake eyes 11 times in a row at a Las Vegas dice table. Possible, yes, but highly unlikely. Formation of the first nucleotides was either a long-shot odds secular miracle or a downright supernatural miracle.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.